and also affects its receptor affinity. So you have to balance the two of those things. So you have to know an awful lot about the molecule to be able to work that out. And we had to do a lot of mutation analysis of the protein to work out which glycosylation sites were important. We actually engineered forms that have a longer half-life, more glycosylation, still bind to the receptor and so lead to longer activity in people. So that's one of the things that we did at Amgen in the past 10 years.
And I don't want to jump into yours if you're going to hit it later, but how long did it take you to develop that drug?
So to develop RNS, which was the new erythropoietin, the basic research took probably 5 or 6 years. The clinical trials took quite a bit longer than that, so it was between 10 and 15 years, I think. And that's generally what it takes. It takes us, on a good decade, it takes us 10 years to get from basic discovery, starting a project, finding out that it works, to having a drug that goes in, that's approved to go into people. And it can be a lot longer than that. So for instance, if you've got a drug where you want to measure hemoglobin as an endpoint, the clinical trial is very short. It's maybe 3 months. You just need to see the hemoglobin grow up their red cell mass. Sorry.
What drugs are you working on currently?
So, I can't tell you the early stuff because we're not allowed to, but I can tell you general areas. One of the things I've been involved with is a protein to mobilize iron in the body. A lot of people who have anemia don't have anemia because they don't have erythropoietin. They have anemia because they've got iron locked up in their bodies that can't be mobilized, and you need a lot of iron to make red blood cells. So the protein that controls that is actually a 25 amino acid peptide. It's a peptide hormone called hepcidin. And so when there's too much iron in the body, the liver makes hepcidin, and hepcidin downregulates iron uptake and downregulates iron release in the body, and so you decrease iron. But as well as being increased by too much iron, it can be increased by inflammation. So there are a lot of people out there with diseases that have inflammation, that have anemia that couldn't be treated. So what we did is we produced an antibody that recognized hepcidin, which is a difficult thing to do because it's tiny and it's tightly folded. We treated mouse models with this, and we showed that it effectively treats this anemia. So that's one of the things that may or may not go into people in the next few years. We're not sure. Right now, we're actually doing some very cool stuff. Have you guys talked about developmental models like zebrafish, xenopus? So you can model diseases in systems like C. elegans, the worm, zebrafish, etc. And a lot of very interesting targets come out of zebrafish research. So we're potentially considering collaborating with some of the world's leading zebrafish labs to come up with aspects of biology we haven't explored before. Do genetic screens to say, gene X causes a disease, but we can't really target gene X. Can we look for something that can compensate for loss of gene X by doing a mutational screen in zebrafish? And that brings us to another gene that maybe we can target. So, we often think of doing things like this in model systems or in cell lines, etc. So those are some of the kinds of things that I'm thinking about right now.
Can you tell them what cell lines are?
Yes. Cell lines can come from any kind of organism. Usually we use human cell lines, and they often come from cancers. So there's a book out right now, and I never remember the woman's name. I always get it, Henrietta, something like Henrietta, don't remember. So there's a very famous cell line called HeLa that came from I think it was a cervical carcinoma. So what happens to cells to turn them into cancer cells? Have you guys worked on this yet?
We talked about cancer, yeah.
So immortalization happens. You have a couple of mutation events usually that mean that the cells can grow when they shouldn't grow. They don't need the normal growth factors to stimulate their growth. They can grow not next to other similar cells, which means they lose something called contact inhibition. Usually when cells do this, they should stop growing.
So you like it when you say things we've talked about.
So there's several key characteristics that define a cell as a cancer cell. So you can take cells out of people, and you can either select them in culture to sort of grow these characteristics, or you can take cells from a patient sample with cancer. And you can grow those cells, and then they grow in culture, and they're called immortalized cells. You can test how cancerous these cells are by injecting them into animals and seeing if they grow tumours. If they grow tumours, I know, but we do it. If they grow tumours, they're called transformed cells. So those are the two levels. But generally, as a surrogate for doing experiments on animals, wherever we can, we do experiments on cell lines. So for instance, for the hepcidin project, when we wanted to see how cells respond to hepcidin, we take cell lines, we overexpress the hepcidin receptor in those cells, and then we add hepcidin to those cells, and we use something to read that out. So we often use things called reporter genes. Have you covered reporter genes? So reporter genes are genes that make a protein that's very easily detected by some means or another. It could be a protein called green fluorescent protein.
Oh, we did that. We've done that. We just didn't know they were called reporter genes.
So green fluorescent protein comes from Achloria victoria. Very, very useful protein because it fluoresces. So
and they're familiar. They did the red fluorescent protein.
Okay. So there's green, yellow, and I think there's blue too now.
Okay.
So they all come from species which fluoresce. You find the protein that causes that, you express that, and upstream of the actual gene, you put regulatory regions that are controlled by whatever you're interested in. And we know enough about the sequences to do that. We can engineer sites that will say, if I add this protein, I'll see this transcriptional response, it will turn on this gene, and how much protein I get from the gene is directly correlated to how much stimulus I put in. So you can use that to screen cell lines and tell when you've got activity from a particular protein. And then if you want to, you can see if small molecules can interfere with that process. And that's how we do screens. There's a slide in here somewhere, actually, that shows what a screen might look like. Okay, I'll get to it in a minute. But you can do this in... You guys have seen 96-well plates? You use 96-well plates?
Yeah.
They're about this big. Well, when we screen for activity of small molecules or proteins, we use 384-well plates, so tiny, tiny, tiny things. You need a lot of automation to read out. So you need readouts that are very easy to process, that essentially machine reading it can say generally yes or no, above or below a threshold. And that's how we decide if small molecules affect a pathway or not. So we engineer the whole system generally to get sort of a perfectly responsive system and to see whether a small molecule can turn that pathway on or off. So I'll revert to the talk a little bit. I talked about recombinant proteins. There are lots of kinds of recombinant proteins we can make. The most common one are antibodies. We use antibodies to neutralize things, and I guess you guys are familiar with that concept. So it's hard to do screens to get really good antibodies because what you want them to be is extremely specific. You learned about the specificity of antibodies. I don't know if you learned that the epitopes that they recognize, that even if there's a tiny bit of conservation in another protein, sometimes you get cross-reactivity. That's what drives autoimmune disorders. When you start to recognize self-proteins and you get cross-reactivity, maybe against something you were exposed to, some illness, etc. So we work very hard, for instance, at making sure our antibodies are incredibly, incredibly selective. And I don't know how much you guys know about affinities, but...
talked about it.
So generally, if you talk about a small molecule recognizing a protein or a protein-protein interaction, they can be nanomolar affinities. Better than that is picomolar. Absolutely, more or less the best you can get is femtomolar. You know, these are log scales, three log scales down each time. And we generally go for picomolar affinity, which is very specific and highly selective. So we have several examples of proteins that we make that are either antibodies, fusions of a bit of an antibody to another protein to increase stability, a peptide fused to an antibody so that the peptide has the specificity and the antibody gives it long half-life because antibodies circulate for a long time in the body. There are lots of combinations of things we can do. And we can take a bit of one protein, stick it to a bit of another protein. That's called domain fusions because the different proteins have different domains.
Are you using the domain? Are you using the antibodies to target to bring a chemical to a cell? Are you using it as a vector? Or?
So now people are developing things called chemibodies that can do that. It increases the half-life because if you think of an antibody as a big Y shape, the bottom bit of it, the Fc region